All right. So, hey, everybody. Thanks for coming to our final Solanaceae seminar for the session. So our speaker today is Vanezza Morales, and she's a geographer from the Pontificia Universidad Católica de Chile, or sorry, she did a master's in science and in biodiversity and taxonomy of vascular plants at the University of Edinburgh. She's worked in research projects on the taxonomy of Chilean plants and biogeography, vegetation and botanical history, and the spatial analysis of those plants using GIS. And so currently she's studying for a PhD in forestry, agriculture, and veterinary sciences at the Universidad de Chile, and she works as an associate researcher at the National Herbarium also. So thanks for being here, Vanessa, and I'll let you take it over and get started.
Okay, thank you very much, Lucas. First of all, many thanks for having me to do this last talk from this year's sessions. I'm very happy to be here because it gave me the opportunity to go back to work we did in 2020, like published in 2020, and we started in 2019. And it was great to go back because I discovered so many things that have changes the what we did. So that is exactly what I want to show you with this presentation. So basically, I'm not gonna talk about the taxonomy that we did in that work. I'm gonna talk about how we did that work and how we solved the problems that we found during the process of doing it. So now I'm gonna share the presentation, which is called, let's see if this works now. Yeah, just one change. Okay, if you can confirm that you're looking like the right presentation, like in full screen. Yeah,
looks good.
Great, thank you. So I call this talk Chisantos, the backstage of a synopsis and the future perspective. So basically the things that we couldn't solve while we're doing this synopsis. And my name is there, Vanezza Morales-Fierro, but there are also two other names, which are the co-authors of the synopsis. And they are not giving the talk, but the things that I'm presenting to you, they were involved on this. So it's a good way to respect their work in this talk. So let's start. Basically, what I want to know first is how I'm going to split this presentation in three parts. The first one is a brief introduction about Chisanthus, where I'm going to tell you how many they are, where are they, and how they look like. The second part is how we elaborate the synopsis. And basically here I'm going to focus on the data sources that we use and the process, how we use them to get the final result. And the last part is the unresolved questions or perspective, things that we should be doing in the future. So starting, I'm going to tell you that Chisanthus is it's considered a small genus within the family Solanaceae. They are all annuals and perennial herbs. According to the two latest taxonomic works, which are from 2020 and last year, 2021, there are 19 taxa. 16 species and 3 specific taxa, intraspecific taxas. Species of Chisanthus are distributed in the south cone of South America, but only in Chile and Argentina. But as you can see in the map, most of the records are in Chile. Just few of the records, which correspond only to species, goes to the other side of the Andes and cross to Argentina. Another important fact about Chisanthus is previous molecular work place this group as the sister group of all the rest of Solanaceae. This means that it's an early branch diverging from the family. About morphology, how Chisanthus looks like. They develop very strong zygomorphic flowers that comes from pentamerous flowers, and the lobes are rearranged into leaves. These leaves, the upper lip is composed of 3 lobes, as you can see in this dissection. So you have a 2 central lobe and 2 lateral lobes. And then you have the lower lip, which has 2 wings and a gill, like in a Papuliniaceae flower. This bilateral symmetry not only affects the corolla, it also affects the androecium and the gynecium. So in the pentamerous flower, you will have 5 stamens, but here only 2 develop fertile stamens. The other 3 are not developed. And the gynecium is bicarpellated. So basically, this same form that is dissected, you can see it in these other pictures, where you can see the upper lip, 3 lobes, like the central one, the lateral ones, which are bilobe or trilobe, and also the wings. And here you can see the gill. Yeah, and the anthers, like the fertile anthers. Because of this shape, these plants, like vernacular names, have similar names over the country, and they are from little bird flower, little butterfly, and poor man's orchid. So this is the brief introduction. I don't want to go deeper into this subject because one of my colleagues already spoke about the synopsis and went through all the species. So now, very quick, we are gonna go to the second part, which is how we elaborate this synopsis. In 2020, we published the synopsis of the Chisanthus. But basically, what we were trying with this work was to clarify or resolve the questions that my co-authors couldn't resolve in 2008, because they were just doing literature research, like examining all publication, original publications, but there were several problems that they couldn't resolve, like the fact that several publications have 2 groups of flowers and they were called the same, but you can see they weren't the same. Like, not the same species, but they were treated with the same name. So this is the reason why we started to look at different data sources that help us to have a bigger picture about the genus and the species. And first of all, we started to work with herbarium specimens, physically and also virtual collections. Almost simultaneously, we work with publications that were found in other different sources like BHL, Google, and, for example, Plant Illustration. I'm going to go deeper in how we use each of those, but in the following slides. And in the last place, we work with photos from fieldwork that we were gathering from different sources on internet. And we did a very good job with that because we have some limitations, like working with herbarium specimens, and I'm going to explain you why that. So first of all, we work with herbarium specimens as most traditional taxonomies. You go to an herbarium and you work with the collections that they have. And here in Chile, there are 2 main herbaria. SGO in Santiago and Conc from Concepción in the south of Chile, and they are the two main herbaria here in Chile. We also work with the private collection of Andrés Moreira Muñoz, who is one of the co-authors of the work. But we also went to visit 3 herbaria in Argentina: Buenos Aires, La Plata, and the Botanical Institution of Darwin Institute. So finally, we got examined 6 collections and more than 1,000 sheets of Chisantos, together with just 6 collections, which is a very good amount of specimens, considering that you have less than 20 species within the genus. Now, going to the virtual collections, we work in the following way. As we didn't know which one were the collections or the institutions that held most of the specimens of Chisanthus, we use GBIF, Global Biodiversity Information Facility. And I'm pretty sure that you know it, and you know that it has a lot of occurrences, more than 2,000 million occurrences from almost 2,000 institutions. So we went here to see how many occurrences of Chisanthus they already have. And the thing is, if you click on the number of occurrences from any taxon, you will see the list of these occurrences on GBIF. But the thing to get the records that have an actual image that you can see and identify is a little bit difficult, because you have to go, you have to click in each of those records to get to that list of data. And sometimes you cannot download the image. So what we did is we use Global Biodiversity Information Facility, GBIF, as a previous work to get to the virtual herbarium or each of those institutions that held those collections of Chisanthus. So basically, we use one of these filters, which is publisher. And here you go. We have the list of publishers that have most of the collections, like the specimens from Chisanthus. Here, as you will see, first place is Phytokis, and I'm gonna go back to this data because it's not worth to mention it now, but you can see one of the institutions that have more records from Chisanthus is Naturalis, Instituto Botánica Darwinian, Missouri Botanical Garden, Naturalis, el Royal Botanic Edinburgh, esto es Smithsonian, I think so, and Museum from Paris, and it goes on and on and on. So getting that information allows us to go to each virtual herbarium of these institutions. So here we have the virtual herbarium Morque, Paris, Naturalis, British Museum, Smithsonian, and Royal Botanic Edinburgh, which were the herbarium that have more specimens digitalized. In total, we examined 24 collections, and we found almost 500 specimens, which is half that we got from other herbarium, like we were visiting physically. But the good thing about this digital herbarium is that allow us to check very old specimens that we couldn't see physically, and they were from Europe. For example, here I have an example of Chisanthus candidos, which is a species that grows in Atacama Desert. And in most of floras and checklist of plants from Chile and from South America. This plant is mentioned to be growing in Atacama and Coquimbo, which are two administrative regions in Chile. In Atacama, we could see a lot of specimens from herbaria, but we failed to find a single sheet that the locality of collection was Coquimbo. So we have to trace from where this data come from. And the thing is, in the original description of the species, they say that this plant was found near wild near Coquimbo by Mr. Bridges under the number 131356. And as I told you, with all the specimens we checked, we just could find the populations of Chisanthus candidos was only in Atacama. So this was a question that we couldn't resolve until we found all the type of specimens from Bridges. There were 6 sheets, but only one was giving us the data, like the full location where the plant was collected. All the others were saying Provincia of Coquimbo. But this one is full. It says hills near the valley of Huasco, Provincia of Coquimbo. The thing is, today, Huasco is not located in Coquimbo. So this is a reconstruction of the map of the old Provincia of Coquimbo, where when Bridges was in Chile. So here is Coquimbo and here is Huasco. And he was right. At that point, Huasco was considered on the Provincia of Coquimbo, but not now. So this is one of those data that we have to check, because administrative units in the countries, they change the limits. So you cannot continue citing species where they actually are not. And this is not one of those examples where you have a very old population that disappears and you cannot find it there anymore. Like he was collecting in the actual province of Atacama. So that was great. But we also have to face some problems during this work. I will say the most problematic thing was that leaves in Chisanthus doesn't work to do identification, taxonomic identification, except for 2 species which grow in Atacama Desert. But from all other species, the leaves are completely, if not same, very, very similar. So the thing is, when you don't have flowers on the herbarium sheets, it's almost impossible to identify the species you are looking at. Another problem that we have with the herbarium specimens was that the flowers lose the color and also the shape during the process of drying and the pressing. So that was very difficult to identify all material or material that have few flowers and they were completely closed, like not open to show their colors or spotted design. It was difficult. Like this is the typical specimen that is in the undeterminate. But going back to JV, it was great to see that after checking all those specimens, we returned our database to JV as a data set. And this is one of the things that I'm very grateful of preparing this talk, is that there are 40 other works or publications that have been using our data to create other studies. So it was great to see that people is using it. And it's not just for Chisanthus, it's also for answering other scientist questions. So it was good. And this is also the reason why phytogeese appears on the list of publishers with Chisantos data, is because we update our list of examined specimens. This is everything about herbarium specimens, how we do it, how we did it, and this stage, it was simultaneously with the search of publications. So now we are going to that step, like that group of data sources. So starting, we work with a website that I'm pretty sure you are familiarized on, which is IPNI, the International Plant Name Index, which is very good for finding original description of the names of the species. And the great thing about IPNI is not they give you the name of the publication. They sometimes, and most of the time, they have a link of another website where you can find the actual publication, like have access to the full document. That website is the Biodiversity Heritage Library, where, as I told you, you can have access to the full document. So you can have the descriptions, illustrations, photograph is there, more recent. So it was great. And during this work, we found all the original descriptions from this website. We didn't have to go further to other sources trying to see that. But the thing is, we were also looking for another kind of publications that provide morphological characters that help us to separate the species. So we were looking publications that were also analyzing the morphology of the species and giving us new morphological characters that weren't mentioned on the original descriptions. And that was the time where we must start to dig in other sources. And one of these sources is plantillustrations.org, which is a not commercial website, and they gather all illustrations which belong to public domain according to European law, and it works very, very easily. They have a search box. You just have to put the scientific name of the vernacular name of the species you are looking for, and they are going to show you all the images labeled under that name. So this works this way. You just go and select one of these images and press there and click there, and you will find a full image of the illustration. But the most interesting thing, they tell you where you can find that illustrations, because usually these illustrations can come with a description and also some notes, because most of them come from horticultural journals. So when we did the synopsis, this wasn't this easy. What I'm going to show you is new. And I'm jealous because it could be so much easier at that point. So if you go to the name of the journal or the book that contains that illustration, sorry, one important thing. For us, working with illustrations, it was very, very important, because within the genus, we have several species that were described from cultivating specimens. Like, they took the seeds from Chile, they went to Europe, mainly UK, and they grow the plant, and then they realized that the plant was new to the science, and they describe it. So if you go to the name of the publication of the book, you will get redirected to the list of volumes of that journal or book. And then if you want to look at that illustration and the associated text, you just have to press again, and ta-da! This is the entire publication. So you can have access to the illustration, the description, like generic description, and also the specific description, and also some notes that were very valuable for us because gave us an idea from the area where the seeds were collected. But sometimes that didn't work. We couldn't find those publications in DHL, so we have to use Google, which was also very useful because they have a small collection of all journals, like British Flower Garden. So sometimes you're lucky enough to find them and to have access to that. For example, in this case, where they were publishing a beautiful illustration of Chisanthus pinnatus var humilis. And this was one of the things that we really used to trying to match things that we could see on fieldwork and also with the illustration. And that is everything about publications. And now I'm going to the third data source, which are the photos from fieldwork. So I would like to start saying that this was a way that we find to trying to counteract the limitations that we find when we were working with publications, illustrations, and also with herbarium specimens. So basically, we went to different sources where we can get images and download it. I'm not gonna go deeper now. I'm just gonna go through all the sources that were more important for our work, and then I'm gonna tell you what we did with them. So, well, the first source, it was In Naturalis, which is an initiative of the California Academy of Science and the National Geographic Societies, where anybody can put an observation of an organism, and then this observation goes to a group of naturalists that can be scientists or not, and have a good knowledge about that organism or the group of organisms. And then they start a discussion about, do they really belong to this organism or to the others? And that is the way that how they are creating a new repository for biological records. And this is not exception for Chisanthus. So you can type on the search box Chisanthus, and that is it's going to take you to the main page of all Chisanthus species. And then from the list, you can pick the one that you are interested on. And that is exactly what I did for this example. So I pick up this species that we call Chisanthus Carlos Muñozi in our synopsis. And if you press there, you will find all these photos that the people have been putting under that name, because they found some similarities between the flowers. After that, we were using also a social media that I'm pretty sure you know how to use better than me, which is Instagram. So we were just using the typical hashtag Chisanthus, or perhaps the full name of the species, and we get all the photos labeled under that name. Sometimes those photos have information about the place where they're seen, the date, and the photographer, it was easier because it was the name or the user of the account. But for example, in Naturalis, most of the times you have those data. Here in Instagram is not always available. But the good thing about Instagram is that you can ask. And most of the times you get the reply from the user. So it's good because it's very valuable data that you can use in to do another things. And the last source that we use to get these photos from fieldworks is a website called Foto Naturaleza, that it works that just for Chile. They charge plants, fungi, animals, and landscape, and it works very similar to eNaturalis because they have a catalog where you can find like the group, and you go and then you have all the species of Chisantos listed here. And then if you go into one of these photos, you will see all the photos that are labeled under the same name. This doesn't mean this is entirely correct. I mean, the good thing is to have that image because you can work with it as it was an herbarium specimen. So finally, what we did with all these images. First of all, we use them to see how the flowers look like along the range, like its distribution, like the entire distribution. For example, all these photos come from the sources that I told you before. So this one is, this goes from north to south. So Cuesta del Melón, Papudos Apoyar, Algarrobo, Punta Endo, y La Campana National Park. As you can see, there is some variability within the color of the corolla. Sometimes the patterns are more diluted, but the spots is still there. So it was good to know that there are stable things and also things that change. But we couldn't see this on herbarium specimens. So it was a great source just to see how the plants look like on the field. The second thing we did with these photos was to create a database where we basically treat the photos as herbarium specimens, and we put the locality date, the photographer, which is the same as the collector, and just did the we compare like the distributions between herbarium specimens and photos and see if they were matching or they were different in some localities. And the thing is, most of the time they were the same. So it was very, very good. Here I'm presenting two examples for Chisanthus grahami and Chisanthus hogeri, which are species that grow in the Andes. And in blue dots, you can see herbarium specimens that we check, and we could put the coordinates. So you can see them in the map. And we did the same, but with the photos, with the red dots. So red dots are localities that we could extract from the photos that we found in different sources. And the last thing that it was very good with this database of photos is that it becomes a very good source for new localities when we go on fieldworks. So when you see this distributional range of a species, you actually are seeing historical range. So you have specimens collected in 200 years ago, but probably that population, it doesn't exist anymore if it's a very populated area. So what we see with the red dots, because they come from photos, is the most recent collections of those species. Because in recent times, most of the people do not collect the specimen like herbarium specimens, but most of them take photos. So it's a good source also to know where I have to go and look for a certain species in present years, like in the present day. And that's it, everything about the sources that we use, and also the process that we follow with all of these sources, how we use them, how we assemble all this information in order to get the synopsis. And the thing is, because we started this work in 2019, we have some troubles to go on fieldwork. So we started to work on Chisantos in April 2019, and on October, it was the time where we should go on fieldwork. Unfortunately, we have a political issue here in Chile that we couldn't go on fieldwork. And after that, at the following year, the COVID arrived, and that was also a very difficult time to go on fieldwork, like waiting for the next season, like spring in 2020. So we just stopped working and decided to send the synopsis to the publisher because we didn't know how much longer the lockdown it's going to last. So now I'm gonna tell you which ones are the questions that we couldn't resolve, or things that we think it would be very good to study within the genus, which is the part 3. So Chisanthus porregens is a very difficult species to identify, because there is a lot of variability within closer populations. So in this map, I'm showing you the color of the corolla, which is also the same color that we use as an outline of these pictures. So basically, these photos, which look like Chisanthus porregens, the illustration of Chisanthus porregens, because that is one of the species that was described from cultivated specimens. So we don't have type specimens. So the match we did, it was between the photos and the illustration. So these ones, which are pink, they seem to grow closer to the coast. So this is the Valparaíso region, this is the metropolitan region, and this is O'Higgins region in Chile. So all of the pink flowers seems to appear closer to the coast in Valparaíso region. And then you have some populations, some individuals which present a white corolla, just with that yellow spot in the middle lobe. But we also found very weird plants, very weird flowers with not dark spots, but just with the yellow spot in the middle lobe that grows in metropolitan region. And here is the difficult thing. These all can be grouped, or at least we group them, or as Chisanthus porregens in our synopsis. But there are also a lot of flowers growing here that exhibit a different type of corolla, like the shape of the lobules are different. Also the design, like the spotted design is different. And further in the south, there is another type of corolla. And we couldn't check this because as I told you, it was very difficult to get herbarium specimens from these plants. Most of them are just photos. And just this year, I started to collect them so I can take measurements, and I can also try to do some molecular work to see if they are together as a lineage or on the same branch of the phylogeny. But this is a group very interesting to work and trying to resolve and see if they are the same species, very variable species, or different species of the genus. The second thing that I would like to show you is about the phylogeny, which was published by Lavandero and its colleague last year. So in this work, they were doing two things, but basically they were describing a new species from the north of Chile, and also the reinstatement of species described by Johnston from the Atacama Desert, like the coast of Paposo, which is very well known as a center of endemism here in Chile. So they were looking at different types of characters, like morphological characters, ecological characters, and also molecular data that support these species. So here they did nuclear and plastid phylogeny that supports that the Chisanthus fayax and the new species Chisanthus nutantiflorus were different from all the other Chisanthus that we already know, which is what's great. And for this work, they were following a previous revision of the genus by Grau and Grombach in 1984. But the thing is, if they follow that work, there are many things that were difficult to understand, because in our synopsis, we were working with the type of flower. Like for us, it was very important to see how the flowers look like color, pattern of like the designs of the drawings in the flowers. And that is a little bit confusing in that work. So, for example, here they have Chisanthus litoralis. Grau and Grombach, between that name, under that name, recognize two types of flowers. This one that they show on the phylogeny, but also this one that we consider that is very different. In our synopsis, these are under the same species, but as two different varieties. And the thing here, we weren't brave enough to split them, because with all the data that we had very similar distributional range, like overlapping range. So some people were saying they grow together, and probably this is just variability within the same species. But now, after going on fieldwork, I can say that I have never seen these two flowers growing together. They have ranges that overlap, but they don't have the same populations. They grow in different places. So it will be very good to see if in another phylogeny we could have samples of this type of flower and also of this one, and see if they are related. And if they are related, are the same or different lineages, or they are completely different. Another interesting thing is that in Grau and Grombach, they proposed the Chisanthus tricolor as a new name, where they put different species that we consider different. And they put them under this name. So these 3 types of flowers, they consider as a Chisanthus tricolor. So when I saw this phylogeny, it was like, which one of these flowers correspond to that sample? I also want to see what happened with the other 2, to see again if they are related or not. The same happened with Chisanthus pinnatus, because this flower that you can, as you can see here, this is the kind of flower that grows in central Chile, but the name Chisanthus pinnatus corresponds to a species that grows in the south of Chile, and they always have purple corolla with these drawings here, which are different from that one. So it's true, they share like the same basic form of the flower. They are little flowers, very slender, but the pattern of the that you can see on the flowers is different. So again, are the same species or not? And finally, Schizanthus porigens. As far as we could see, Schizanthus porigens look like this, but Grau and Grombach considered that this type of flower was under Chisanthus tricolor. So also it would be good to know which kind of flower it's related to Chisanthus litoralis. So as you see, as in terms of phylogeny, there are a lot of new data to get into it to see if these different points of view about the species in Chisanthus are true, or we have more species to split. Another interesting thing that we did some work on, but we stopped it because we don't have enough data, is to assess the conservation assessment for all the species of the genus. So at this point, we have worked just with one species, which is Chisanthus litoralis, because we have a lot of work there, and we know very well the area where the historical populations were growing. So basically what you see here in the map is in blue dots are the populations that no longer exist. We have been there and they are not there. And the red ones are the localities that we have visited and we haven't found the species, or we have found the species, but it's experiencing some changes in the land uses. So it's very likely it's going to disappear. And as you see, like the historical records of this species, it's a huge area in the coast of Chile. And mainly that is the problem of this species. All these areas are being used now as residency and also for cultivation. So there is not much space for natural population in these areas. That is the reason why we participate of the process of evaluating this plan, and finally it got categorized as vulnerable. And the good thing about publishing and doing this work is that I started to receive a lot of records, like from photos, like, hey, I found this plant in this small locality in a beach near, don't know, Papudo. And we went to see them. and it was great to see that the plant actually was there. So we have been updating this map. I think it continues with as a vulnerable, but it's good to know that there are some places that we still having those populations and we didn't know. The fact is that communicating this to people, they start to look in different places for these plants because they are beautiful. So they really want to see them and they really want to find them. And when they do that, they just write to you and tell you, hey, the plant is here. But unfortunately, that is not the case for all the species in Chisantos. So for example, here we have the distributional map for Chisantos pinnatus, which is made of historical records from almost 200 years ago, more than 200 years ago until 2019. The thing is, how many of this population exist today? Can we really assess the category of conservation, the conservation category of this plant using these records? We don't know if all this population is still existing. So that is the first question that we should answer. And the second one is, what are the threats? What are the threats of these plants? Because we know little about it. Because of the area, like this is a typical area of forestry in Chile. So I could think that they will be affected by the replacement of natural vegetation and that we cannot find them in plantation, like pine plantations. But the thing is, doing some propagation and also some collections, there are some data that started to appear that it seems that Chisanthus needs some disturbed areas to grow. So this is unpublished data that I'm sharing with you because a very good friend is working on it. So he has done some experiments the last 3 years with seeds of Schizanthus litoralis in a small area in a school in Concon in Chile. And he has noticed things that we didn't know about Chisantos. For example, like there are animals that eat the plants before they flower, actually get flowers. So he, at the beginning of the season, when you throw the seeds, you can see like small plants started to grow out of the exclusions, but when you start to have very green leaves, they just disappear. So you have to put some care on build this exclusion from the animals. He has also been working with thresholds of water, like trying different kinds, so like amounts of water to see when the seeds started to germinate. And he thinks that if you put some water every week between 2 months, you will have a long season of flowers and without problems. Like every week, putting some water, it will be the most effective thing to do, like 2 liters. And he also was looking at different levels of growing of the plants when the substrate is disturbed. So I'm gonna go back to the other picture because I want to explain this. So he was putting some seeds here, and you can see small plants with many flowers, but I think there were 3 individuals inside of this exclusion. But the thing is that he didn't put any seed on the exclusion next to it, and he was just removing the substrate and watering these plants, like this area, as the same as the other one. And he got this amazing amount of flowers, just removing the substrate. So it's interesting to see how can you see different results, not managing the amount of seeds, just removing the soil. And that is something that it could explain why these species are growing or are found in disturbed areas like forestry plantations, roadsides, gravelly slopes that are very difficult for other type of plants, but they seem to grow very happily there. So this year he found a new population of this plant next to a new road that they built between 2 cities in the coast, in between Santiago and Concon. So something is happening with Chisantos that appears to like this kind of perturbations. And I think this is the last thing that I had prepared, like about new perspectives or things that we should work on in Chisantos. So just tell you many thanks for being here. And if you have any question, I'm very happy to answer if I know how to answer.
Awesome. Thanks, Vanezza. That was a really cool talk. Yeah, if you have questions, you can either just type your question in the chat or type in the chat that you have a question, and I can call on you and you can turn on your mic and ask a question. Get comments streaming
just
in
comments, and I'm very
so
grateful
far.
of them. Thank you very much.
All right. Any questions? So far not. I think you answered everybody's questions already.
I know this is not the typical talk that you could see in Sol seminar, but
oh, no,
I thought it will be useful because we face these difficult questions like, what can we do with these plants that were described from cultivation, and we couldn't have access to herbarium specimens, and we didn't have a clue how they look like. And actually we learned a lot doing this work, and I hope this helps someone to continue doing some research on Chisanthus or repeating this in other groups of Solanaceae or another families.
Yeah, definitely. I thought it was great how you integrated so many different sources of information, and I'm sure that that'll be helpful to other people who have similar problems. All right, well, we're at the hour mark, so I guess if there's no questions, we can go ahead and call it. And thanks for presenting again. That was really great. And yeah, thanks for being. Oh, actually, it looks like there's a question.
Yeah, there is one question. So what about searching in living collections? Well, here in Chile, I don't know living collections. Probably they do exist, but I don't know them. And as I told you, we started this work in a very difficult time in Chile between political issues and COVID. So it was difficult to move from one place to another. And because I was studying at the Royal Botanic Garden in Edinburgh, I knew they were not Chisantos there. And we also have very good friends at Kew. So I didn't know about living collections of these plants in abroad or here in Chile. But if there are, I'm interested in continue working in Chisantos. So if you know a collection that I should be visiting, please tell me and I will be happily to go.
Yeah, and anybody who couldn't make it to the talk, if it'll be on YouTube. So if you do know of any collections, let Vanezza know. Looks like there's another question. With the available information, is it possible to assess the species regarding threats or classification for conservation?
This is a question that we always discuss with Andres, because he is the co-author of the synopsis and he's really keen about completing those assessments. And the thing is, as I told before, we have the historical range of distribution, and following the guidelines of the IUCN, you can have some idea of the conservation status of that species. But the thing is, you cannot complete that if you don't have the traits. If you don't have if you have not identified the problems that are affecting those populations, you cannot complete like the assessment. You can just go and say, okay, look, you have this range, and according to the square meters or kilometers that this plant is distributed, you have, don't know, a possible vulnerable category, but it's not completed. It's very difficult. You have to do a lot of field work to see if there is, which are the threats for each population.
Cool. All right. No other questions? Okay. Well, yeah, thanks again for presenting, and hopefully see you at the future Solanaceae seminars.
Hopefully.
So, yeah.
Thank you very much, Lucas.
Yeah. Have a good.
Thank you everyone for being here. Have a nice afternoon or evening.
Yeah, you as well.